Hello, good morning or good afternoon from wherever you're watching in the world. My name is Brittany Gessentine, and today I will be presenting a brief overview of cancer. So starting off, I want to talk about how prevalent cancer is in the United States. So in 2018, there were 1.8 million people that were diagnosed with cancer at some point in their life. And in 2017, there was a 1.6 million occurrence of diagnosis in the United States. So it is very apparent that cancer is very prevalent in the world we live in. It has affected multiple people in my family, friends of family. And as the statistics are showing, as we go throughout the years, there seems to be a large increase in cancer, even though we spend so much money annually trying to eradicate this disease. So let's start off with the basics. What is cancer? So cancer is a genetic disease. It occurs when the cells in the human body begin to divide and they do not stop. Cells lose contact inhibition and apoptosis, necrosis, and autophagy of old and damaged cells. The apoptosis, necrosis, and autophagy stop. So it stops breaking down these old cells and getting rid of them, and they begin to form mass clusters. Contact inhibition occurs when cells basically sense the environment around them. So when a cell has contact inhibition, it can touch up against another cell and say, okay, we need to stop growing. We've reached our maximum spread. So example, if a cell was in a petri dish and you put several epithelial cells right next to one another, as they continue to grow and reach the edges of the petri dish, they'd say, okay, it's time to stop growing. However, with cancer, this loss of contact inhibition causes great divides. So in that petri dish example, the epithelial cells will begin to stack on top of one another and thus will eventually climb over the sides of the petri dish. So due to these losses, these tumors can be formed and eventually metastasis can occur if the conditions are right. So this leads to the next question: how does cancer form? So there's a variety of different ways that cancer can form, but we're going to talk about the three main ways today, since this is just a brief overview. These are topics that we focused a lot in our textbook throughout the semester, and the three topics that are most discussed in cancer peer-reviewed articles. So mutations in genetic code form due to DNA damage. This can be induced by carcinogens. Such carcinogens that we've talked about throughout the semester are environmental carcinogens, tobacco products, cigarette smoking, certain chemical products that are used. The list goes on of carcinogens, and every day there's new carcinogens that are being added to this list that people once didn't think as a carcinogen. So that can cause mutations in genetic code. Radiation can also cause double-stranded DNA breaks. So in UV radiation, it can cause skin cancer due to UV, due to double-stranded DNA breaks due to radiation. In addition, viruses can also cause DNA damage. Viral vectors can enter the human immune system and can be introduced to DNA replication if certain checks and balances are not in place to prevent an invasion. So the genetic mutations on chromosomes that happen in particular are deletions, insertions, substitutions, duplication, inversions, and reciprocal translocations. So in deletion insertions, this is when certain aspects of the chromosome are either taken away or added. Substitutions and duplications, in substitutions one swapped for another, and in duplications, of course, one aspect is duplicated. Inversions and then reciprocal translocations are more, I wouldn't say rare, but less common than the deletions and substitutions and insertions. basically they swap between the chromosomes. It's not on one chromosome, like substitutions within different aspects of the DNA molecule. It's within two different pairs of chromosomes. So what mutated factors can lead to cancer? So the first I want to talk about are proto-oncogenes. Proto-oncogenes in their normal form regulate normal cell growth, and when they become mutated, they're called oncogenes, which more people are more familiar with. They can lead to the development of cancer. So typically, oncogenes have a gain of function. They code for growth factors, receptors for growth factors and hormones, intracellular signal transducers, nuclear transcription factors, and cell cycle control proteins. Mutations to these specific functions result in the overproduction of growth factors, large amount of replication signals, uncontrolled stimulation of pathways, and increased transcription factors leading to cell growth. So this increase leads to cancer development and tumor formation due to there being a lot of an increase in cellular promoting factors within the environment. So the mutation of these cells can lead to that tumor formation. In addition, tumor suppressor genes are used to regulate the cell cycle. When the cell cycle is not arrested and cells are able to move too quickly throughout the cell cycle, masses of cells conjugate due to the lack of regulation, and cancer can form. Normally, tumor suppressors inhibit cell proliferation, and this is a loss of function, whereas which is opposite of what the oncogenes are. So when the oncogenes are mutated, they have a gain of function, but mutated tumor suppressor genes result in a loss of function. And in order to have a loss of function in these tumor suppressor genes, there has to be two mutations of the same two mutations of the same suppressor gene on the same type of gene must be defective. So you have to have two matching sets to completely have it as a loss of function. Some examples of these tumor suppressors are the RB gene. The RB gene regulates the G1 checkpoint within the cell cycle. Inactivation leads to cell proliferations and increase. In that G1 checkpoint, it's not looking as closely at the cells to make sure that they're ready to progress. They're just kind of quickly moving them through. P53 is another example of this. P53 arrests the cell cycle to allow for DNA to be repaired, damaged DNA to be repaired. If damaged cells cannot be fixed due to P53 inactivation, apoptosis, necrosis, and autophagy do not occur due to the mutated and damaged genes. So this allows damaged genes to continue throughout the cell cycle and continue to wreak damage to the other cells that it surrounds as it goes through DNA replication. In addition, mutations to sensor and effector kinases. Sensor kinases, detect damage in the double helix DNA, and if they detect this damage, they automatically autophosphorylate themselves and lead to a plethora of downstream effects to effector kinases that again leads to more downstream effects that leads to DNA repair. If the kinases are mutated, DNA repair can be altered, which can lead to cancer formation. In this mutation, the effector kinases are no longer seeking out DNA damage. Or it could be that the effector kinases are mutated. So the sensor kinases are sending signals to the effector kinases, and the effector kinases are kind of going, well, what am I going to do about it? So that is a quick way to lead to cancer formation. So I just want to read off this little hierarchy that I made. It's just a quick review of what we just went over. So in normal cells, they function properly. They have contact inhibition. They repair themselves as normal, no problem. They continue to move through the cell cycle as needed. But when DNA-damaging agents such as chemicals, radiation, and viruses damage this DNA, it needs to go through repair. So again, in successful DNA repair in healthy cells reverts back to its baseline. However, failure of DNA repair can lead to the mutations in the genome of somatic cells, which we briefly discussed. Activation of growth-promoting factors and activation of tumor suppressor genes and kinases lead to unregulated cell proliferation. Alterations in genes that regulate apoptosis obviously decrease apoptosis, and necrosis and autophagy, which are also used to eliminate cells that no longer function properly. So this unregulated cell proliferation and decreased apoptosis leads to clonal expansion. This is where, and we haven't discussed this yet, but we will as in the next section of this. Once this clonal expansion occurs, angiogenesis and escape from immunity and additional mutations can lead to tumor progression, which leads to malignant neoplasms, and that leads to invasion and metastasis. So tumor progression and metastasis. Tumor progression and invasion occurs when tumors are allowed to grow. Tumor microenvironments can play a large role in tumor progression due to their supportive nature. In our textbook, we went over tumor microenvironments and how they can help and how they can prevent the invasion of metastasis. But since we're talking about the development of cancer, I just want to focus on how they can be supportive in this example. So of course there's increase in angiogenesis, which, as we know, is the increase of blood vessels and vasculature to these cell clusters, which gives it the nutrients that it needs to continue its growth. Increased in cellular immune response can also goes hand in hand with this angiogenesis and allows for tumor progression and formation. So you have an increase in swelling, you have a lot of an increase in inflammation, you have a lot of cells going to this site to help either attack or to help progress whatever may be occurring. In addition to that increased vascularization. We also see, as I briefly discussed before, is the escape from immunity. So essentially the body will it will attempt to fight off the cancer briefly, but then it moves to a point where white blood cells are no longer effective in fighting off the tumor formation. Natural killer cells can no longer do their jobs properly. Macrophages no longer help digest the cells. So we do see that escape from immunity at that point. Evasion metastasis occurs when epithelial to mesenchymal transitions take place and break through to surrounding tissues, blood, and lymph, allowing it to spread to different parts of the body. So we briefly talked about this in our textbook about the epithelial to mesenchymal transitions. This occurs when these cells transition from one cell type to another, and they can actually break through the basement membrane due to anchoring and invasion. So they can break through the basement membrane and can impede to surrounding cell types. If, for example, if it is skin cells, it may break down into the dermis and deeper into the arm, for example, if it was on the arm. So it can also go into the blood. We hear a lot of like lymphomas, cancer in the blood. We also hear of it spreading to lymph. So when it spreads to lymph, it can travel throughout the rest of the body, since lymph is used to help filter in the body. One interesting example that I read about breast cancer metastasis when we were doing our research on our organ of choice. I had the pancreas, and as I was reading through, there was an article about breast cancer metastasis, and there was something about the article that just caught my eye. So when I opened it and went through it, they were discussing how they check for breast cancer metastasis, and that they will actually remove lymph nodes from a section of the armpit that has the lump in the breast. So example, if it was your right side, they would remove some lymph nodes from underneath your arm and check it for metastasis. They can also use special dyes. They can inject it into the tumor that is in the breast, and they can then look to see if the dye, had traveled to lymph tissue. So I thought that was interesting. That's not something that I think is very common knowledge, especially in cancer diagnoses. And that's the next section of this, is the detection and treatment. So diagnostic and cancer screenings can include biomarkers. We talked a lot about biomarkers this semester. Depending on the type of cancer you have, there's specific biomarkers that can be found within blood serums and within different liquids, such as like bile. You can check bile for specific biomarkers. In addition, nucleic acid analysis and genetic screenings. For example, like the breast cancer that we used in this example has BRCA1 and 2 genes that put you at a greater disposition to developing breast cancer. They have tissue sample analysis, like I discussed in the breast cancer, where they remove the lymph, or they actually remove part of the tumor to determine the functionality of the tumors and its cellular pathology to determine if it's been corroded or not. In addition, diagnostic imaging such as CTs and MRIs are also used to determine if tumors have formed. In my example of pancreatic cancer, the tumor formation of pancreatic cancer is not well known. It's not seen. It's not diagnosed until very late stage. It has a lack of symptoms, and it has a lack of a bump. Usually in breast cancer or other cancers, you'll feel a lump, but you can't do that with the pancreas due to the visceral fat that we have. So it's very interesting to see how these biomarkers can play a large role in diagnosing cancer. Of course, out of all 3 of the examples, the biomarkers, nucleic acid analysis, tissue samples, and diagnostic imaging, testing with biomarkers is probably going to be the easiest to do. It's going to be the most cost effective. If you had a patient that doesn't have insurance, a CT, MRI, tissue analysis, and even genetic screenings can be very costly to them, whereas a basic biomarker panel could be a couple $100s. So that is definitely one thing to keep in mind during diagnostic thoughts. Is the cost effectiveness. If you've known anybody that has cancer or know of a friend or family, you know how costly it can be even with insurance. And I think that the introduction of biomarkers is going to be greatly served in this field. Lastly, I want to talk about the treatment of cancer. So of course you have surgical removal of the tumor. That's usually the first step. They want to get that tumor out of there so it quits spreading. And hopefully they catch this before metastasis has occurred. So in breast cancer, they're going to go in and they're going to do a mastectomy. They're going to remove as much of that tumor as they can possibly get out of there. In addition, chemo and radiation therapy are also the two leading treatments for cancer. So in chemotherapy, of course, they use chemotherapeutic drugs that target cancer cells to kill them, and then in radiation therapy, they use a beam of radiation to kill those cells. Sadly, with both of these treatment options, there is a decrease in the overall outlook of life. The the way of living becomes decreased. Due to these treatments, they make you very sick. You don't feel good at all. You know, I've known of patients and people in my family that have bone pains because of it. So overall, it just decreases the outlook of life, how they're living. However, there are very promising treatment options that are being researched and discovered every day. Some of these examples are therapeutic drugs. These drugs can affect growth factors and their receptors. So once the mutation occurs, we can go in there and start doing these drugs to help reverse those effects. Drugs that target the immune system to decrease vascularization to tumors, to decrease angiogenesis, to decrease the immune response that occurs. Drugs that target gene replacements and gene knockout via oncolytic viruses. So I don't know if this is relatively new, but I feel within the last few years I've learned a lot more about this than I have ever learned during my whole undergrad and graduate studies. But we're introducing viruses to the human body in order for them to go through DNA replication, in order to replace broken genes. In addition, lastly, for treatments, interferons to regulate anti-tumor growth and increase apoptosis. So this, of course, the tumor will have already been formed, but this can help decrease that tumor formation. So hopefully within the next, I would say 5 to 10 years, hopefully we have a breakthrough in science where we're able to apply these techniques that we're learning, the therapeutic drugs that are being tested right now and the interferons. Hopefully that we can use these in everyday medicine and practice to decrease our overall numbers of 1.8 million and 1.6 million of diagnosed cancer patients. I think that as we go along, we're going to run into a problem where we're going to introduce more carcinogens than we have treatments. So now is the time to definitely be seeking out those other opportunities. So that is all I have for today. I will link all of my sources either in the email or in the post below, whichever one will upload the best. If you have any questions or want me to follow up with anything that I may have said that I may have not been clear on, please just shoot me an email. I will be more than happy to do that. Thank you so much for listening.